Hi students, welcome to the session of Biochemistry Recall AnySet. As far as the AnySet biochemistry questions are concerned, what I feel is that 3 or 4 questions were direct repeat from the previous AIMS exams, and the topics that were asked is almost the most repeatedly asked topics only. For example, like vitamin B12, which is a very commonly repeatedly asked topic. The same was asked in the AnySet as well. And as usual, it's a trend of AIMS exam, like the previous AIMS exam, is the trend was to ask 1 or 2 questions from the basic chemistry, which was seen in this time also in the biochemistry session, where you can see a question on normality and molarity. So that is what I felt, the same pattern or the trend of AIMS was followed in the AnySet as well. And as the PGI JIPMER exams is also in the same exam, what I felt is that a little more importance was given to the molecular biology techniques in the genetics topic. So we will see the questions one by one. The first question is number 1. All are true regarding telomerase except it has reverse transcriptase activity. Yeah, of course, it has reverse transcriptase activity. It's a true statement. Only found in eukaryotes. Yes, it is found only in eukaryotes because telomerase is an enzyme activity that is present in those DNA which are linear DNAs, and the eukaryotic DNAs are the linear DNAs. So it is present in the eukaryotes. It's a true statement. It maintains the chromosome length. Yes, it is also a true statement. Now, active in the somatic cells. That is a false statement here, because the telomerase enzyme is usually not present or not active in the somatic cells. So the answer here is D. So we will be discussing about little bit in depth about the telomere and the telomerase, because in the 20th edition Harper, a new chapter is added on the telomere disease. So I will be discussing the telomere and the telomerase based on the Harrison 20th edition. First of all, we know what is end replication error. In the video session of telomerase, I have clearly mentioned about what is the end replication error. Shortly saying about the end replication error, it is, when the DNA is getting replicated, we know that the primers, so this is the 2 strands of the DNA, the template strands, and you can see this is the primer, and on the primer we know that the new strand of DNA is synthesized in which direction? In the 5' to 3' direction. So this is the 5' and this is the 3'. And in the other also we can see that on the primer the DNA polymerase is extending. Only thing is that it is not seen in the same manner. One is continuous, the other is discontinuous, which I am not mentioning here. So you can see it is replicated, it is extended in the other DNA on the template strand as well. So once finally when everything is over, we know that in the daughter DNA or the daughter strand that is synthesized, there is no primers, right? RNA is not there, which means that the RNAs are removed. So when the RNA is removed, the ends of the chromosome, or in the case of human DNA, we know that's a linear DNA where at the ends of the chromosome, these primers cannot be replaced because the DNA polymerase cannot function on a scratch or a vacuum. It is not able to synthesize a DNA strand, which means at the end of the chromosome, once the primers are removed, there is an error that is called as the end replication error. So what I have highlighted here is the end replication error, where you can see the 5' end of the daughter strand, there is a gap. Or in other words, I can say that the 3' end of the parent strand, so this is the 3' end of the parent strand, these are the 3' end. So these 3' end of the parent strand is not replicated. So what happens after every cell division? Normally, after every cell division, there is a loss of about 52, that is after every cell division there is a loss about 50 to 200 base pairs. 50 to 200 base pair is lost after every cell division. So what will happen? So a pictorial representation of what is happening if this is happening after every cell division is that you can see that this is the ends of the chromosome. What are the ends of the chromosomes called as? The ends of the chromosomes are called as, so these are called as the telomere. So you can see the telomere is, you can see there is a shortening of this telomere after every cell division. You can see this is almost, the telomere is almost lost. So this is what is happening after every cell division. So this phenomenon is called as telomere attrition. So this phenomenon is called as the telomere attrition. You can see this is the telomere attrition. This is the molecular basis, or this is a normal physiological process, and this is the molecular basis of what is called as the Hayflick limit. I'll explain what is Hayflick limit later. So this there is telomere attrition. This telomere attrition is the molecular basis of the Hayflick limit, and this is the basis for aging. So this is why we all are getting aged, because of the telomere attrition, and you can see progressive shortening of the chromosomes. Okay, so once we have learned this much, we are going to learn what are the consequences of this telomere attrition. The consequences of the telomere attrition, number one is there is limit to the cell division, which means the cell division, the same DNA is getting divided every time. So when the cell divides, the template strand is the parent strand itself. So after every cell division, there is progressive shortening, and the cell division stops after a certain extent. That is what is written as, when the cell division, several cell divisions is happening, the cell division will be stopping. So there is a limit to the cell division, which means the cell is not able to divide further. Then there is limit to the cell proliferation. So there are two limits which is happening because of telomere attrition. One is the limit to the cell division. The second is the limit to the cell proliferation. Okay, so if that happens, what will happen? What is the next consequence of it? When the chromosomes is getting shortened and the proliferation is limited, then the body has a tendency to repair it, because our body is always like that. So what will happen as a result of this? This will activate the DNA repair mechanisms in the body, which are the normal DNA repair mechanisms only, because the body or the cell is finding that the telomeres are getting shortened, or the chromosomes are getting shortened. So which is prevented by activation of the DNA repair mechanism. So when the DNA repair mechanisms is activated, it is actually adding chaos further, because it is again making things complicated in the way that when the DNA repair mechanisms are activated, it again causes certain effects, or what is the consequences of this activation of the DNA repair mechanism. By the activation of the DNA repair mechanism, there is end-to-end fusion. You can see there is end-to-end fusion, where you can see the chromosome ends is getting fused. Then there is breaks in the chromosome, as a result of this there will be breaks in the chromosome. Sometimes one chromosome can be missed, that is leading to aneuploidy, and there is chromosome instability. So totally, even if you forget everything, at least you remember the last consequence, chromosome instability. So progressive shortening of the chromosome, activation of the DNA repair mechanism, which lead to chromosome instability. This is the result of the telomere attrition. This can happen in almost every cell, but it will not happen in certain cells where there is an angel. What is the angel? To protect the ends of the chromosome. Or, that in a shoelace, there is a tip, right? If the tip is intact, that the shoelace will be intact. Otherwise, what will happen? These thread will get degenerated, so that you can easily separate each and every threads of the shoelace. So just like that, there is something, or there is a cap at the ends of every chromosome, which is actually protecting the ends of the chromosome, and there is an enzyme also there. So we are going to learn about what are the protective mechanisms in the ends of the chromosome. The angel which protect the telomeres, number one is the telomerase complex. So what we all know is the telomerase complex. Discuss about the telomerase complex later. So before that, we should be learning about something more which is present at the ends of the chromosome. They are called as, certain proteins are seen at the ends of the chromosome. They are called as sheltering complex. Okay, so the protectors of the ends of the chromosome, number one is the sheltering complex, which is certain proteins, specific proteins that is present at the telomeres. They are called as the sheltering complex. So first we learn about the sheltering complex. What is the function of the sheltering complex? The function of the sheltering complex is it inhibits that DNA repair mechanisms. So, you know that because of these activation of the DNA repair mechanism, the chromosome end-to-end fusion, instability, everything is happening. So, what happens is that it inhibits. So, function of the sheltering complex is inhibit the DNA repair mechanisms. DNA repair mechanism. So, which will prevent the degeneration of the chromosomes. That is the action of sheltering complex. Apart from the sheltering complex, we will be learning about one more complex that is called as the telomerase complex. So what are the components of this telomerase complex? You can see that in this picture, there are a lot of proteins. Don't learn everything. I'll just tell you the salient features. Let's just learn that. So the telomerase complex is having certain proteins which is called as number one, TERT. What is TERT? TERT is the telomerase complex is having a reverse transcriptase activity. So TERT stands for the telomerase reverse transcriptase. Reverse transcriptase. Then the second is TERC. TERC. The TERC is actually this enzyme, telomerase complex, is having an RNA template, and that is called as TERC. So TERC is the RNA template. RNA template. Then this telomerase complex is having a protein whose name is very interesting. That's why I have picked that name here. That is dischirin. So there is a protein there in the telomerase complex, that is dischirin. Then one more component also I find it interesting. That is about TCAB. TCAB1. What is the role of this TCAB1? The TCAB1 is the protein which is helping in the trafficking of the telomerase complex. So that is what is written here. TCAB1 helps in the telomerase trafficking. You know, trafficking means it is helping in the carrying of the telomerase complex. So this telomerase complex, it is carried to some place. Which is the place, or what is the destiny of this telomerase complex? You know, the telomerase complex should bind to the telomere, right? So for the telomerase-telomere interaction, these complexes or TCAB1 is the one which is helping in the trafficking of the telomerase enzyme to the correct location. The telomerase enzyme or the telomerase complex is trafficking. It is carried with the help of this TCAB1 to certain nuclear structures. So what is this nuclear structure? The nuclear structure is called as the Kajal bodies. It's called as the Kajal bodies. So inside the Kajal body, so this is the Kajal body. Inside the Kajal body, which is a nuclear structure, inside this nuclear structure, the interaction of the telomere, because the telomere has to bind with the telomerase enzyme. So the interaction of the telomere with the telomerase enzyme is taking place inside the Kajal bodies. So that is what is shown here. So this is the Kajal bodies. What is shown here is the telomerase complex, and what is shown here is the telomere. So what is shown here is the telomere. So the telomere is interacting with the telomerase inside the caudal bodies. For that, what is helping is the TCAB1. So, in short, in the telomerase complex, what we have learned is, number one, it has a reverse transcriptase enzyme. So, that is called as TERT. Then, it has a TERC. So, TERC, where there is an RNA template. So, that is why this telomerase complex is a ribonucleoprotein. Ribonucleoprotein. Why? Because it has protein component as well as the ribonucleotide component, because that is an intrinsic, or it is an RNA template which is seen inside the complex itself. So, it is having RNA plus the protein. So, that is why it is a ribonucleoprotein. Then the dischirin and the TCAB1, these are all parts of the telomerase complex. Now, the third component which is protecting the ends of the chromosome is called as RTEL. What is RTEL? RTEL is a DNA helicase activity which is present at the ends of the chromosome, which is not a part of the telomerase complex. Shelterin is different, telomerase complex is different. The third thing which is protecting the ends of the chromosome is called as RTEL. RTEL is a DNA helicase. It's a DNA helicase. And what's the function of this RTEL? It is the one which ensures the telomere length. So, it is ensuring the telomere length. So, these is the protectors of the ends of the chromosome. Now, I'll just summarize how the telomerase is acting in our at the ends of the chromosomes in the eukaryotes. So, what we have learned is the telomere are the ends of the chromosome, and the ends of the chromosome is having, the telomere is having a peculiarity. That peculiarity is the telomere end is always having a hexanucleotide repeat of TTAGG. So, there are about 100 to 1000 hexa means 6 nucleotide repeat. So, that is a repeat of TTAGG. So, it is a hexanucleotide repeat. Hexanucleotide repeat is, there is a repeat of 6 nucleotides, and that is TTAGG. So, you can see this is a repeated. It is repeated at the telomere. So, telomere is the end of the chromosome. It has around 100 to 1000 hexanucleotide repeat, which is the TTAGG repeat, and the telomerase enzyme is different. So, don't confuse between telomere and the telomerase enzyme. Okay, coming back to how this telomerase act. So, we have seen that in the initial picture I have shown that what is end replication error. End replication error is the 3' end of the parent DNA is not replicated, or there is a gap in the 5' end of the daughter strand. So, this is prevented by the telomerase complex. So, I have told you what are the components of the telomerase complex. I hope you remember TERT, TERC, TCAB1, dischirin, etc. Okay, so this is the telomerase. This is the telomerase enzyme. You can see this is the telomerase enzyme, and what you are seeing here, what you are seeing here is the RNA template, which is a part of the telomerase complex itself. You can see this RNA template is complementary to the hexanucleotide repeat. That is, this is always, the telomere end is always TTAGG, etc., TTAGG, etc. So, the RNA template is complementary to this TTAGG. What will happen? With the help of this RNA template, the telomerase enzyme can bind to the ends of the chromosome. So, that is what is shown here. You can see the telomere is binding. The telomere end, you can see the telomere will bind to the telomerase enzyme will bind to the telomere with the help of this RNA template, and you can see it is adding the reverse, because with the help of this reverse transcriptase activity, you can see on an RNA template what is being added. You can see the deoxyribonucleotides are being added. So, that is why it is having an RNA template, and what is happening here is a reverse transcriptase activity. Reverse transcriptase activity. So, you can see the DNA is being added. Now, once the 3' end of the DNA of the parent's DNA is extended with the help of this. So, what you can see is the 3' end of the parent's DNA is getting extended. So once the 3' end of the DNA is being extended, what is the next step? So you can see the 3' end. So this was the 3' end of the parent DNA which is being extended, and what happens later on, what is happening is normal DNA replication process with the help of DNA polymerase. So this is a DNA polymerase. This is a DNA polymerase. You know the DNA polymerase, first an RNA primer will be synthesized as usual. RNA primer will be synthesized and the DNA polymerase will extend this. This is the 5' to 3' direction, right? So you can see the DNA polymerase will extend, or it will extend that RNA primer. So just like what is happening in the replication, don't complicate things. So we can see the 3' end is extended and the DNA polymerase will come. It will come, a primer will bind, then on that primer DNA polymerase will add the deoxyribonucleotide in this direction. So initially what happens is there is extension of the 3' end. So 3' end of the parent DNA is getting extended, and what is happening on the other strand is after some time, or after the extension of the 3' end, the normal DNA replication, that is adding of primer, then DNA polymerase will extend that segment, which means that the shortening of the DNA can be prevented, because even if that end primer is removed, it is removed from the already extended DNA, which means the shortening of the DNA can be prevented, which is normally happening in all the cells after every cell division. So where is this telomerase and activity is present normally in our body? So where is this telomerase activity present in our body? The telomerase activity is present in the stem cells, which all stem cells, adult as well as the embryonic stem cell. Then it is present in the lymphocyte. It's present in the lymphocyte and majority of the cancer cells. Majority of the cancer cell, it is present. So what are the pathological effects which is happening because of the germline mutation that is happening in the telomerase complex? So as a result of germline mutation in the telomerase complex, what happens is normally telomerase activity is absent in the somatic cells. So I have shown you that it is present only in the stem cell, majority of the cancer cell and the lymphocyte. It is absent in the somatic cell. So what will happen if there is a germline mutation in the telomerase complex? It results in a disease which is called as telomere disease, otherwise called as telomeropathies and the telomere spectrum disease. So I'll just show you what are the salient features of this telomeropathies. The first manifestation is in the skin. In the skin, it causes a condition which is called as dyskeratosis congenita. Dyskeratosis congenita, it is actually a triad. This triad consists of reticular skin pigmentation. It consists of reticular skin pigmentation, dystrophy of the nail dystrophy, and oral leukoplakia. I'll show you the picture. The same picture that is given in the Harrison. You can see this is the reticular pigmentation. This is actually hypopigmentation. So you can see reticular hypopigmentations. You can see this is the oral leukoplakia and you can see the nail dystrophy. So this is the manifestation in the skin. Going down, you can see there is a manifestations in the other organs as well. Next is in the bone marrow. What is the manifestation in the bone marrow? It results in the aplasia of the bone marrow. So it results in bone marrow aplasia. Now in the CNS, it causes everywhere, most of the site it causes aplasia. So in the CNS, it causes cerebellar. That word is important. It is cerebellar hypoplasia. It causes cerebellar hypoplasia. Cerebellar hypoplasia. And there is a name that is given for that cerebellar hypoplasia in Harrison. It is also called as Hoyral-Riederson syndrome. So cerebellar hypoplasia, which is seen in the telomeropathies, is called as Hoyral-Riederson syndrome. Now in the lungs, it causes fibrosis, pulmonary fibrosis. Pulmonary fibrosis. pulmonary fibrosis, and in the liver it causes non-alcoholic steatohepatitis and cirrhosis. Steatohepatitis and cirrhosis. And in the eyes it causes trichiasis, tear flow obstruction and exudative retinopathy. So that is trichiasis, tear flow obstruction and exudative retinopathy. And this exudative retinopathy is also called as Revesz syndrome. It's also called as Revesz syndrome. Okay, so that was about the telomere and the telomerase. I have covered a little bit more extensively on the telomerase because this was a new chapter added in the latest Harrison. That's why I have described a little bit more in the telomerase question. Now we move on to the next question, the DNA methylation. The DNA methylation is not involved in the this is an exact repeat question of June 2020 AIMS, but unlike that time, it was actually not a multiple answer type of question. It was a single response type, a single answer type of question. But here it is a multiple answer question. Multiple answer is there in this time in the INI set. But the question and the topic is exactly the same. So we'll see the question. DNA methylation is not involved in chromatin remodeling. Definitely there is a role for DNA methylation. It is chromatin remodeling. It is a true statement. It is involved. Then RNA splicing. No, it is not involved in the RNA splicing. Gene silencing. Yeah, it is an epigenetic modification which causes the inhibition of transcription, which is actually the gene silencing. So it is a true statement. Then base excision repair. No, there is no involvement for DNA methylation in the base excision repair. It is a false statement. So the question here is not involved in. So it is not involved in the RNA splicing and base excision repair. So the answer here is 3, that is B and D. So I will just show you about this DNA methylation and the chromatin remodeling. So this is also from the latest Harrison. A few words about the DNA methylation, which is newly added also. You can learn that. DNA methylation, we all know that it is DNA methylation takes place in which nucleotide residue? It is taking place in the cytosine residue. So it is taking place in the cytosine residue at the CpG dinucleotide. So that CpG dinucleotide, I've already explained about the CpG dinucleotide in the epigenetics video. You can see that. If there is a DNA methylation, what is the net result of this? It causes condensation of the chromosome, or what you can say as the chromatin condensation, which is forming a heterochromatin. So heterochromatin means it is a repressive chromatin which will not allow the transcription factors to bind. So there is less amount of transcription. So it is repressive chromatin, and definitely it leads to gene silencing. So DNA methylation is involved in the gene silencing. Now what is the enzyme for it? This is what I want to add in the DNA methylation. We have learned that the DNA methylation enzyme is DNMT. So that is DNA methylation, DNMT. There are different isoforms for DNMT, which I want to stress here, which is added in the 20th Harrison. DNMT1 is there, then 3A and 3B is there. So DNMT1, what is the role of DNMT1 is the DNA methylation after replication and transcription. So DNMT1, the role is methylation of the DNA after replication and transcription. After replication, the newly synthesized strand is initially not methylated, but later it is getting methylated. So that is the role of DNMT1. Now what's the role of DNMT3A and 3B? 3A and 3B is the one which is methylating the CpG island. So we all know DNA methylation CpG island, which is the enzyme DNMT also we know, but which is the correct enzyme? It is DNMT 3A and 3B, which is involved in the methylation of the CpG dinucleotide. And where is the CpG dinucleotide present? It is present not in all the regions of the gene or in the regions of the DNA. It is present in the transcribed regions and the intergenic region, which means the region between two genes. Okay, so the intergenic regions and the transcribed region, the CpG dinucleotides are present. Okay, so along with it, I just want to add about what is called as a TET enzyme, which is also given in the 20th edition of Harrison. So TET enzyme is actually which reverse the methylation. These are the enzymes which reverse the methylation which is normally present in our body. The TET enzymes, the numbering is 1 to 3. So TET enzymes 1 to 3 are there. What's the role of the TET enzyme? The TET enzyme progressively oxidize the 5-methyl cytosine. When the DNA is getting methylated means the cytosine is getting methylated, and these methylated DNA are progressively oxidized and these methyl groups are removed. How? So the role of TET enzyme is the 5-methyl cytosine is oxidized progressively to 5, initially to 5-hydroxymethyl cytosine. So 5-hydroxymethyl cytosine, then to 5-formyl and 5-carboxy cytosine. So 5-formyl and 5-carboxy cytosine, which means that methyl group is not there, which means TET enzyme, TET enzymes reverse the DNA methylation. Then already I have discussed this in the video of epigenetics, that is what are the inhibitors of the DNA methylation, that is the drugs which are used as inhibitors of the DNA methylation. The inhibitor of DNMT are 5-aza-deoxycytidine, 5-aza-deoxycytidine and decitabine. Then inhibitors of HDAC. HDAC is what? HDAC is not something which is involved in the DNA methylation, but that is the enzyme which deacetylases the histones. So histone deacetylase. This is again another epigenetic modification which I have covered in the video of epigenetics. So inhibitors of HDAC are the varinostat and the valproic acid. So valproic acid and varinostat. So I have discussed about the DNA methylation of all and added points to what we have already discussed in the epigenetics videos. What is chromatin remodeling, or what are the processes involved in the chromatin remodeling? We have to see whether DNA methylation is involved in that or not. So this is given in Harper, the latest edition of Harper. The chromatin remodeling includes histone modification like histone acetylation, histone methylation, etc. Then DNA methylation. So it is a part of chromatin remodeling, DNA methylation. Then the nucleosome, chromatin remodeling also involves a process called nucleosome repositioning, remodeling and displacement. So that is something related to the nucleosome. Nucleosomes can be displaced, nucleosomes can be repositioned, and the nucleosome can be remodeling. So these are all involved in what is called as the chromatin remodeling. I'll show you the picture of the chromatin remodeling. This is given in the Harper. So what is the role of this DNA methylation or methylation here? You can see this is a chromatin. You can see these are all nucleosome, and you can see this is the promoter region. So this data, it's a promoter, and you can see these are all the transcribed regions. So these are the transcribed regions. So initially what you are seeing is the transcribed regions are all nucleosomes, right? Which means they are all condensed, right? So what happens if what is being added here is the ATP dependent chromatin remodelers is being added to these structure, and what happens now? You can see now these nucleosome which was there in the INR and the DPE, which are the downward promoted element and the initiator sequence, which are all transcribed regions. So now you can see the nucleosomes has disappeared, which means the DNA has become less condensed, which means it's a formation of what? It is a euchromatin formation, right? Euchromatin, it is euchromatin. Now you can see the RNA, this transcription complex, this is the transcription complex or the RNA polymerase and other transcription factors, which is now binding to the promoter region, and the DNA is getting, or the gene is getting transcribed. So that process whereby you can see there is in this case what you are seeing is less condensation by the binding of chromatin remodelers. So I'll just show you what are the components of these chromatin remodeling, so that I will just want to specify or clarify that methylation is involved in the chromatin remodeling. So what is given in the Harper is that ATP dependent remodelers. So that is what is given here. ATP dependent remodelers will covalently modify the histones. Using the enzyme histone acetylase, and that is HAT. HAT is the histone acetylase and methylases. You can see methylase. So methylation is a process that is involved in the chromatin remodeling. Now there was one more option, base excision repair. Is there any role for DNA methylation in base excision repair? There is no role for DNA methylation in the base excision repair. I'll just summarize what is base excision repair. You can see there is a mutation where there is a single base change in this particular DNA. So what happens is there is an enzyme which is acting here, which is called as a glycosylase enzyme. When the glycosylase enzyme bind, what happens is that nucleotide, nucleotide is a base plus sugar plus a phosphate. So from that nucleotide, whatever is the base is being removed. So that is what is called as the base is being removed. Now once the base is being removed, then comes the action of an endonuclease which is called as AP endonuclease. So first enzyme is glycosylase enzyme which cut the base. Then, so from the nucleotide that base is being cut. So once the base is being cut, it becomes if it is a purine base, it becomes a purinic side, right? Then what is acting is an enzyme which is called purinic endonuclease. So that is AP endonuclease. So you can see the AP endonuclease is acting. Now what happens? That complete nucleotide is now removed. Now what happens? The polymerase, whatever, it creates a defect here, right? So that is filled or that is repaired by the DNA polymerase. So you can see this is repaired by the DNA polymerase activity. So this is summary of the base excision repair, where you can see the base is excised with the help of glycosylase. Then that sugar and the phosphate is also removed by an endonuclease activity. Then that defect is corrected using a DNA polymerase enzyme. So this is base excision repair. Is there any role for methylation here? No, there is no role for methylation in the base excision repair. So we have seen the question number 2. Now I'm going on to the third question. The third question is a basic question which is usually asked for UG entrance exam. That is about respiratory quotient. Told you now, this aims always have it's prefer something they'll ask about basics. So respiratory quotient is what is called as it is a ratio of carbon dioxide released to the oxygen used. So we'll see the question first. The oxygen utilized is 250, carbon dioxide liberated is 200. So out of the 250, 200 is liberated. What is the respiratory quotient? That is the question. So the answer, so how to find out the RQ? The formula to calculate the RQ is respiratory quotient is volume of carbon dioxide produced to volume of oxygen consumed. So in that case you can see both are volumes, right? So there is no unit for respiratory quotient. So in the given example, it is the volume of the carbon dioxide produced, that is 200 divided by 250. So that comes to 0.8. So the answer here is 0.8. Now I'll just mention about what are the respiratory quotient of different macronutrients. Number 1 is the carbohydrate. So when the carbohydrate is being oxidized, we know that by complete oxidation, we know that carbon dioxide is released. So if I am considering about glucose, the formula of oxidation of the glucose is the C6H12O6, that is the glucose. The glucose when combines with 6O2, it produces 6CO2 and 6H2O. So what is the RQ of carbohydrate? So this is the RQ of carbohydrate. So the RQ of carbohydrate is volume of carbon dioxide produced to volume of the oxygen used. So that is 1, right? So it is 6 by 6, which is 1. So it is 1. Now anaerobic glycolysis. In anaerobic glycolysis, is there any oxygen used? Is there any oxygen consumed? No. The carbon dioxide is produced but no oxygen is consumed. In the denominator, the 0 will come, right? So the answer, in the case of anaerobic glycolysis, we can only write it as something like this, right? So there is no, we cannot say a particular, like in the case of all glycolysis, it is 1. No. In the case of anaerobic glycolysis, it is not 1, it is infinity. Now about fatty acid oxidation, the formula that is given here is the oxidation of fatty acid in the case of 16 carbon fatty acid. What is that fatty acid? Palmitic acid. So when a palmitic acid is oxidized, 23 oxygen is used and 16 carbon dioxide is produced. So what is the RQ here? The RQ here is 16 divided by 23. 16 divided by 23. So that comes around 0.7. So RQ of fat is 0.7. Now coming to the protein, there is you cannot say it's very complicated to write the formula of the proteins, but just learn what is the RQ of protein. So if you are eating proteins, the amount of carbon dioxide produced to the oxygen consumed, that is respiratory quotient, is coming around 0.8. 0.8. Now going to the mixed diet. So mixed diet, we cannot say what is the carbohydrate, but roughly estimating, it comes around 0.8 to 0.85. 0.8 to 0.85. That is the respiratory quotient of a mixed diet. Now about alcohol. What is the respiratory quotient of alcohol? Respiratory quotient of alcohol is 0.6. It is 0.6. Now very frequently asked another question and another doubt is, if we are eating only carbohydrate diet, exclusive carbohydrate diet, then what is the respiratory quotient? The respiratory quotient is 1. Exclusive carbohydrate diet, the respiratory quotient is 1. Now excess carbohydrate diet, again another confusion. In the case of excess carbohydrate diet, we know that excess carbohydrate, Harper is saying excess carbohydrate is going for glycogen synthesis. Glycogen synthesis. So if it is going to glycogen synthesis, the respiratory quotient is 1. It remain 1 only, or even less than that. That is what is given in journals and other books. It is 1 or even less than 1. But if the excess carbohydrate is going to fatty acid synthesis, if it is going to fatty acid synthesis, then respiratory quotient is more than 1. Respiratory quotient is more than 1. So as long as, if a question is asked, what is the RQ of excess carbohydrate diet? As long as it is not given, it is not going to fatty acid synthesis, take it as going to glycogen synthesis. So if it is going to glycogen synthesis, it is 1 or even less than 1. If it is going to fatty acid synthesis, then it is more than 1. Along with it, so we have learned about the RQ of carb, fat, proteins and the alcohol. Then going to the organ. So which will be the organ with the maximum respiratory quotient? So that will be the organ which is depending exclusively on carbohydrate, because that is the one which is having maximum respiratory quotient. So organ with the maximum respiratory quotient is the brain. Brain. Why? Because the brain is depending on glucose. It is depending on glucose. Okay. The next question comes, what about RBC? RBC is also depending on glucose in the fed, fasting, in all the stages it is depending on glucose. Then is it an organ with a maximum RQ? It is actually a controversy, because in RBC it is anaerobic glycolysis. So anaerobic glycolysis, we cannot say particular number because it is infinity, because it is divided by 0, which is it's a number which cannot be defined. So we cannot say that as that is the case there, we cannot say that is the organ with maximum RQ. But the maximum RQ will be for brain, because the brain is using only carbohydrates. So these are the things that you have to learn in the respiratory quotient. The next question is a simple question, but the options seem to be a little bit complicated. That's all. The correct order of steps in the polymerase chain reaction. We know the essential steps or the basic steps of a polymerase chain reaction. And we know there is the DNA. This is the target DNA. The target DNA is first denatured, right? Now, once it is denatured, the next step is what? The next step is to this denatured DNA. The primer is being added, right? The primer is added. So if this is the 3' end, this is a 5' end, this is a 3', this is a 5' end. The primers are added to which end? It is added to the 3' flanking region. The primers are added here. And then what happens? It will go to the next step, extension. You can see this is the primers here, and it is being extended. It is being extended in the 5' to 3' direction. 5' to 3' direction. Okay. Now the steps we have seen. The first step is what? Denaturation, right? Denaturation, which takes place more than 90 degrees Celsius. The second step is annealing. So what is happening in the annealing is, we know that the primers are being added. So you can see the forward and the backward primers. So primers is added. So this is the point I want to discuss. You know the temperature is 54 to 60 degrees Celsius. 60 degrees Celsius. In the step of annealing, actually what happens is the primer is hybridizing with the 3' end of the flanking regions of the DNA. The primer, step is annealing. You know, normally, usually in the steps of PCR, we give the name annealing, but the process that is happening during annealing is actually hybridization. It is hybridization. So there is a role for hybridization in the PCR. That is what I want to stress. And the next, there is extension. So this last step, I don't want to describe, because you all know what is extension. You know the primers are being extended, and this is taking place at around 72 degrees Celsius. So now we'll go to the question and see what's the answer. The correct order of steps in the PCR is denaturation, annealing, elongation, hybridization. No. Elongation, no, never elongation will come in the first, so it's also not right. Denaturation, annealing, extension. Yes, it is correct. Annealing, extension, denaturation. No, it is not like that. Denaturation, annealing and extension. That is the correct sequence. So C is the answer, not D also. So hybridization is something that is given just to confuse you. Or in the step of annealing, actually hybridization is the process that is taking place. But usually in the step we will never say hybridization, but hybridization is a process that is taking place. I am accepting that fact, but in this question the best answer here is denaturation, annealing and extension. Okay, now the last option you can see, denaturation, annealing, elongation or extension plus hybridization. No, hybridization is not coming the last. So in the sequence of annealing, if it was given, then it was right, but it is given as the last option, so it is not right. So annealing and extension. So usually in PCR we will never say the word hybridization. We usually say it as annealing, but the process that is taking place there is hybridization. I just want to stress that point. Otherwise, it's a very simple question. The fifth question: the post meal with a high thermic effect is seen in. This is also very repeatedly asked question. The answer here is protein. So we are discussing about what is this post-meal thermic effect. You know, when we eat the food, it is digested, absorbed, and through the blood, it reaches the different cells of the body. In the carbohydrates, the carbohydrates, if it is liberating energy, what is the energy that is liberated? 4 kilocalories per gram, right? In the case of proteins also, it is 4 kilocalories per gram. Lipids, it is also 9 kilocalories per gram. So this is the normal amount of energy that is released. Now, even if the energy that is released is this much, we are not actually getting that energy as a whole, because the body requires some energy for certain processes which is taking place in our body. So, which are the areas where the energy that is generated from these nutrients are consumed? So, first is the resting metabolic rate. So, RMR, that is this RMR is actually it is taking around 10% of the total energy expenditure. So it is taking around 10% of the total energy expenditure. And what is it used for? The RMR is used for the basic metabolic functions in our body. In the sleeping also we are respiring, the circulatory system is working, so all the vital functions. So this is for the vital functions of the body, like respiration, the blood flow, the ion transport, the cardiac activity and all. So that is resting metabolic rate, and which comes around 10% of the total energy expenditure. Then what is the next level where the energy is used? That is called as the thermic effect. The thermic effect is also called a specific dynamic action. Specific dynamic action or thermic effect, or it is also called thermogenic effect. Thermogenic. Okay, so certain energy is used for what is called as a thermic effect. So what is this thermic effect used for? This is the production of heat when the digestion and absorption is taking place. Whatever food we are eating, it should be digested and absorbed. So for the digestion and absorption also we need energy, and that is called as the thermic effect of food. So production of heat by the digestion of food is called as the specific dynamic action, SDA, or the thermic effect. So which macronutrient is using maximum energy when it is being digested and absorbed? It is around 5 to 10% of the total energy expenditure, whereas fat is around 0 to 5%. Proteins is consuming about 20 to 30%, which means maximum energy is used for the digestion and absorption of proteins. So this will be the maximum. Then comes the carbohydrates. The third comes the fat. So this is what is called as the thermic effect. So which is the food that is producing the maximum thermic effect? It is the protein. So don't get confused between the thermic effect and the energy that is released. For example, I've told you in the beginning, the energy that is released per gram is 9 kilocalories for fat, whereas proteins it is 4. So in that case, the fat is the maximum. But thermic effect, if it is coming, for proteins it is the maximum. So what are the other areas where the energy is consumed? That is for the physical activity. So the third thing where the energy that is released from the food is consumed is for the physical activity. You know that it is highly variable depending upon what is our activity. So generally it is used for specific dynamic action. It is used for the resting metabolic rate, that is the RMR, as well as for the physical activity. So this is about the fifth question. Now moving on to the next question. There is alternate splicing of a gene which have segment 1, 2, 3, 4, 5. You have the primer W, X, Y and Z. It yields two products. 1, 3, 4, 5, 1, 2, 4, 5. Okay, so this was a very difficult question for you. I will just make things very simple here. This is actually about the designing of a primer. So if you design a primer, so that we want the segments, see what we want is a product B, that is 1, 2 and 4, 5. So how we can design a primer? Already there is given what are the primers that is used, W, X, Y and Z. So you can see that the primer that is used is, so to find out, what you can do is convert this linear DNA to a circular DNA. So like this you have to draw. And draw the segments here. Segment 1, 2, 3, 4 and 5. Okay. Now we have to draw the primers that is given. So you can see segment 1, there is a primer in this direction, that is W. Then for segment 2, you can see there is a reverse primer, that is X. Now for the segment 3, you can see a primer Y moving in this direction. Segment 4, you can see there is a Z which is moving in the same direction. 5 is not having any primer. So this is what I got from the recall session from the students. So if you see this, you can see that what we want is 1, 2, 4 and 5. Okay, so what is the primer for? If you observe this, you can find that if we want, what we want is 1, 2, 4 and 5. So we don't want this, we want 1, 2, 4 and 5. So for that, this is Z. Okay. So what we want is, usually for getting 1, 2, 4 and 5, we need the primers which include this 1, 2, 4 and 5. So we want two types of primers. You know, if you want a amplification or the splicing, and then it has to be amplified, right? So if you want to splice the 1, 2, 4 and 5, we need two primers. One is a forward primer and the next is the reverse primer. So we see, just cutting that segment 3, see which are the primers here. We have the primers here as the W, X and Z. So what we want is a forward and a reverse primer. This will act as one primer and Z will act as another primer. You can see we don't want this W. We don't want this W. With the X and Z itself, what are the segments that we are getting? We will be getting the 1, 2, 4 and 5. But I doubt whether this is in this sequence itself. Actually, the sequence will come like this. The sequence will come like this. 4, 5 and 1, 2. So if we are using X and Z, what we'll get is 4, 5, 1, 2. W cannot be included here. See, W if you are using, it will include, see if the W is, it will include the 3 also, right? But we don't want that 3. So if we want to exclude the 3, what we want is two primers which is situating or which is going only for 1, 2, 4 and 5. So the Z will take care of this and X will be in the opposite direction. So the primers that we want here is X and Z. X and Z. The options I got is WX, WY, WZ, WXY, etc., which I don't feel that this is the correct recall. If there is an option XZ, that is the answer. So XZ is the answer. So this is how you have to find out the answer for the primers. What you have to do is you make it into a circular DNA and just draw all the try to imagine. You cannot draw on any paper, right? So try to imagine the primers in the position and just find out the forward and the reverse primer, which can include the segment which is the product that is asked. So in this you can clearly see that X and Z can manage for the 1, 2, 4 and 5, which will exclude the segment 3. Now another question is a clinical question. A patient presented with a pallor and cyanosis. Which of the following condition? So this is a clear case of knuckle hyperpigmentation. The knuckle hyperpigmentation is seen in vitamin B12 deficiency. It is seen in vitamin B12 deficiency. But that word knuckle hyperpigmentation usually not given in foreign books, because knuckle hyperpigmentation is not usually seen in white population. It's usually seen in Caucasians. So that is why this knuckle hyperpigmentation is usually not given in the books which is done by or written by the foreign authors. Okay, so this is a case of vitamin B12 deficiency. Knuckle. So we'll discuss a few words about the knuckle hyperpigmentation. Knuckle hyperpigmentation is usually seen in certain ethnic population. You know that knuckle hyperpigmentation is seen. Then characteristically, one endocrine disorder where you can see the knuckle hyperpigmentation is Addison's disease. It's very characteristic. If you search for knuckle hyperpigmentation in Harrison also, what you will get only is the Addison's disease, not vitamin B12 deficiency. I've told you now, because it is not usually seen in the whites. It is usually seen in the Caucasians. That's why it's not given in those books. Thyroid disorders. It is seen. Hemochromatosis, it can be seen. And vitamin B12 deficiency. Okay. So 80% of vitamin, see remember 80% of vitamin B12 deficiency, there is knuckle hyperpigmentation. So it is a very characteristic finding. In isolated folic acid deficiency, there is no knuckle hyperpigmentation. It's exclusively seen in vitamin B12 deficiency. It's very, very important. And 80% of vitamin B12 deficiency, knuckle hyperpigmentation is seen. So not usually seen in folic acid deficiency. And that is what one more thing that we have to concentrate is, or that I want to highlight is, it's seen only in the Caucasians. Then what are the causes for knuckle hyperpigmentation? This is from journals only, PubMed journals. What I find is that the causes that is given for knuckle hyperpigmentation is, one is reduced glutathione. This lead to activation of tyrosinase enzyme. And that is why there is knuckle hyperpigmentation. That is one reason that is given. Possible reasons only. Then defect in melanin transfer between two sites, that is melanocyte and the keratinocyte. So that is also given as a reason for knuckle hyperpigmentation. So this is one journal from which I could find about the knuckle hyperpigmentation, and there I just see one more thing. That is, apart from knuckle hyperpigmentation, what you can also see is the palmar hyperpigmentation on the palm. So you can see here very small letters, but I will just write it here for you. It is the hyperpigmentation in the palms. Hyperpigmentation in the palms. This is also seen in vitamin B12 deficiency. Now going to the next question. This is actually a simple basic +1, +2 standard chemistry question. The normality of 2 molar HCl is dash as 1 molar H2CO4. The answer here is same. What is the relationship between normality and molarity? So normality is molarity into molecular weight divided by equivalent mass. That is the relationship between the normality and molarity. So normality is molarity into molecular mass divided by equivalent mass. So this molecular mass divided by equivalent mass is given by that is a letter N. So that N stands for molecular mass divided by the equivalent mass. And what is this molecular mass divided by equivalent mass? A simple way to calculate that is, if you want to find out the normality of an acid, then just find out how many hydrogen ions are there in the acid. Then that will be the N. For example, if it is the normality, that is, if in the case of HCl, it is 1. So that N is 1, which means 1 molar HCl and 1 normal HCl will be the same, right? Because both are 1, right? Now go to the H2SO4. H2SO4, the N is what? The N is 2. 2 hydrogen ions are there. So that N is 2. So 1 molar HCl and the question here is, you can see here, 2 molar. 2 molar HCl and 1 molar H2SO4. 1 molar HCl and 2 molar HCl and 1 molar H2SO4. So 1 molar H2SO4, when it is converted to the normality, what is it? It is into 2, because the hydrogen ion is 2 there. So they are same. So 2 molar HCl and 1 that when we are comparing it with the normality, it is the 2 itself. So that means it is same. So it is same. So the answer here is same. Now one more N I will tell you here, that is the H3PO4. So just see H3PO4, it is 3. So this is the simple way to relate the normality and the molarity. The next question is enzyme with highest catalytic efficiency, and in the options the Km value and the Kcat is given. So when these two things are given, how can you find out the catalytic efficiency? The formula to calculate the catalytic efficiency is Kcat divided by Km. So it is Kcat divided by Km. So we just have to divide and find out which is the one that is having maximum magnitude. So the options here, number 1 we will be seeing, the first is 10 micromole and the Kcat is 20 per second. So it is Kcat by Km, right? So in the first case it is 20 divided by 10. That is 2. In the second case, the Kcat is 50 divided by, now remember this is not micromole. If you want to compare, all the units should be the same. So instead of nanomole, we have to convert it into micromole. How can we convert nanomole to micromole? You have to multiply it by 10 to the power of minus 3, which means you have to cut 3 zeros here to 10 to the power of minus 3. So you have to cut 3 zeros here. So that is 2. It is coming to 25. Now the next is 200 divided by 2, that is 100. Now the last option is 200 divided by 4, which is 50. Okay, so which is having the maximum? The maximum is for C, that is C is the answer. So this is how you have to find out the catalytic efficiency. So a few words about what is this Kcat. The Kcat is Vmax divided by the total enzyme concentration, that is ET, right? ET. What is this ET? Total enzyme concentration. We know that if an enzyme is acting on the substrate, we know that enzyme, this is a free enzyme, combines with the substrate to produce the enzyme substrate complex, which later on produce the enzyme plus the product. So this is what is happening. So the total enzyme concentration means this is the free enzyme, right? This is the free enzyme. This is the enzyme which is bound with the substrate. So if we want to calculate the total amount of enzyme, we have to combine these two, E plus ES. So this E plus ES is, that is a free enzyme plus the enzyme substrate complex. It is called as a total enzyme. So Kcat means it is the maximum, what you can say, the maximum at the maximum velocity by using all the enzymes. What is the efficiency of the enzyme? So Kcat is something which is what you say, the superlative degree. And the Km is what? Km is, you know that it is a substrate concentration at half maximal velocity. So if we divide the Kcat divided by Km, you will get the catalytic efficiency of the enzyme. Now, we all know Michaelis-Menten equation, that is Vi is equal to Vmax into S divided by Km plus S. I am going to add this Kcat also into this Michaelis-Menten equation. That is, we have seen that the Kcat is equal to Vmax divided by ET, where ET is the total enzyme concentration. So what is the Vmax here? It is Kcat into the total enzyme concentration. So the V is equal to, instead of Vmax, we can write Kcat into total enzyme concentration into S divided by Km plus S. So you should be knowing all these things, because if we have to calculate the initial velocity, but Vmax is not given. So but what is given is Kcat and the total enzyme concentration. How can we calculate the Vi? So just substitute this Vmax here and we will get this. So this is the relationship between the Vi, initial velocity, Kcat, the total enzyme concentration, Km and the substrate concentration. Okay. So the Kcat divided by Km is giving you what? The catalytic efficiency. Same question was asked in previous aims also, but it was not the calculation. What was asked is the maximum catalytic efficiency is represented by, or it is given by Km by Kcat, Kcat by Km, etc. That was the option. So there the answer was Kcat by Km. So in this question, in this time, what is asked is a calculation of the same thing. So the topic is still the same. Now again another question on vitamin B12. The vitamin B12 as far as concerned is very important vitamin that we have to learn everything. So this is another question that is asked in the biochemistry part. In vitamin B12 deficiency, which of the following reaction does not take place? So if you want to answer such question, you should be knowing what is the coenzyme role of the vitamin B12. So vitamin B12, we know that there are two active forms, the adenosyl B12 or Edo B12 and methyl B12. These two are together called as the carbamide enzyme. Carbamide enzymes. The methyl B12 is required for methionine synthase. Methionine synthase. This is also called as homocysteine. homocysteine methyltransferase. Methyltransferase. Okay. Now go to the adenosyl B12. Adenosyl B12 is required for methyl malonyl coenzyme mutase. So these are the two enzymes like methionine synthase and methyl malonyl coenzyme mutase. These are the two enzymes which require vitamin B12 in humans. But there is one more enzyme that is given in books, that is leucine aminomutase. Leucine aminomutase. Okay. So the question here is, if there is vitamin B12 deficiency, which action of enzyme will be low? So for that, it's also a multiple response type of question, multiple answer questions. That is, you can see methionine synthase, definitely it is correct. Methyl malonyl coenzyme mutase, it is correct. Homocysteine synthase, no, it is not homocysteine synthase, it is methionine synthase. What is methionine synthase is also called as homocysteine methyltransferase. Then folate reductase, no. So the answer here is A and B. So you can see here A and B is the answer. So if there is vitamin B12 deficiency, what is accumulated? So you can see here there is accumulation, increased homocysteine. Homocysteine, why? Because homocysteine is not getting converted to methionine. There is increased methyl malonic acid. Methyl malonic acid. So both this will be increased in vitamin B12 deficiency. For folic acid deficiency also there is increased homocysteine. Folate deficiency also, or B9 deficiency also, there is increased homocysteine. Why? Because folate is required for methionine synthase. Because we have seen the conversion of homocysteine to methionine. The methyl group is given by N5 methyl THFA. That is converted to THFA, which is taken by the B12 and it becomes methyl B12, which means for that reaction B9 and B12 both are required. Whereas for the reaction which is called as the methyl malonyl coenzyme mutase, only B12 is required. So B12, if there is deficiency, isolated B12 deficiency, then you can find it out with increased methyl malonic acid. Now the next question is, which of the following enzymes are involved in gluconeogenesis? So this is very careful when you write this question, because it's a very simple question. Everybody will make this question right. But remember that is, if we'll see the option and then I will explain why I am saying that little bit cautious when you answer such questions. The options here is very simple. One is pyruvate carboxylase. Definitely it is involved in gluconeogenesis. Phosphoenol pyruvate carboxykinase. It is not pyruvate kinase, it is phosphoenol pyruvate carboxykinase. Yes, it is also involved. Hexokinase, it is not involved. Pyruvate kinase is not involved. Glucose 6-phosphatase. Yes, glucose 6-phosphatase is involved in gluconeogenesis. So you can see that these are all the key enzymes of gluconeogenesis. Suppose if there was an option called glyceraldehyde 3-phosphate dehydrogenase, will you give this right or wrong? It is right. Why? Because this is a reversible enzyme of glycolysis. Reversible enzymes of glycolysis is common to both glycolysis as well as gluconeogenesis. That is why I said little bit care should be taken to answer such questions. Even though it looks to be or it seems to be simple, sometimes this kind of twisted options will be there. So remember, it is not an enzyme only for this glycolysis. It is involved for gluconeogenesis as well. Okay, so we'll just see what are the key enzymes. You are all familiar with the glycolysis and the gluconeogenesis. I'll just tell you which are the irreversible step of glycolysis. The irreversible steps of glycolysis, number 1 here is the hexokinase bar glucokinase. Then comes the phosphofructokinase 1. Then if you go down, all the other steps, what are given as blue, all these steps are reversible enzyme. Means it is common to glycolysis as well as gluconeogenesis. But what I have written in the yellow letters or yellow highlight, these are all irreversible enzymes. So now last here it is the pyruvate kinase. So these are the irreversible enzymes, whereas if you go to the gluconeogenesis, these irreversible enzymes are bypassed by pyruvate carboxylase, phosphoenol pyruvate carboxykinase. All these steps, all the other steps I am showing is common. It is there in gluconeogenesis as well. Then fructose 1,6-bisphosphatase, and going last it is the glucose 6-phosphatase. So these four enzymes are the key enzymes which is not common between glycolysis and gluconeogenesis. So this is what is about that question number 11. Now moving on to the next question. Ketosis is common in type 1 diabetes mellitus due to. So we will be seeing the what is happening in this type 1 diabetes mellitus or diabetic ketoacidosis in type 1 diabetes mellitus. Then we will go to the options. Okay, in diabetes, type 1 diabetes, we know that the insulin is not produced. So that is the first block, which means the glucose in the blood is very high. You can see the blood glucose level is very, very high. That is the blood vessel, the blood glucose level is very high. And we will be seeing what is happening in every organs. Once the blood glucose level is high, but at the same time, this glucose is not able to enter into different cells, especially the insulin dependent organs where there is insulin dependent glucose transporters. For example, in adipose tissue, it is not possible. In skeletal muscle, it is not possible. In skeletal muscle, it is not possible. So, what is happening is the blood glucose level is rising because it is not able to enter into these peripheral organs where these organs are the one which is drinking the glucose. So, what we have seen is the blood is loaded with glucose, but the cells is lacking the glucose. So, the signal that the cells are getting is that to produce more glucose. The organs that produces the glucose is the liver and the kidney. And what is the pathway that is called, which produces the glucose? It's gluconeogenesis. So what happens here is, there is increase. So what is shown here is the gluconeogenesis. That is increased gluconeogenesis. So there is increased gluconeogenesis. And apart from that, there should be some source for the substrates for gluconeogenesis. What are the substrates for gluconeogenesis? That is, one is, you can see alanine is coming, right? The skeletal muscle, its pyruvate is converted to alanine. This alanine and other gluconeogenic amino acids goes to the liver and it is converted to glucose. In the adipose tissue, there is something that is happening is, we know that an enzyme which is called as hormone sensitive lipase. This hormone sensitive lipase is actually inhibited by insulin, right? In the absence of insulin, what happens? There is excess hormone sensitive lipase. So there is excess hormone sensitive lipase which is degrading the or hydrolyzing the triacyl glycerol to glycerol plus fatty acid. So what's the role of this glycerol and the fatty acid? The glycerol is one gluconeogenic substrate. So it goes to the liver and it is getting converted to glucose. Then what's the role of this fatty acid? This fatty acid is undergoing beta oxidation. So in the absence of insulin, the fat is getting mobilized. There is beta oxidation. It is converted to acetyl coenzyme A. Okay. Now this beta oxidation is the one which is producing or giving ATP for gluconeogenesis. ATP for gluconeogenesis. Now this acetyl coenzyme A can enter into two routes. What are the two routes or possibilities? The acetyl coenzyme A can go for TCA cycle as well as for ketone body synthesis. So two routes are there for acetyl coenzyme A. To which route the acetyl coenzyme A is entering? We have seen that there is excess acetyl coenzyme A, right? So there is excess acetyl coenzyme A, and this acetyl coenzyme A cannot enter into the TCA cycle. Why? Because of the beta oxidation, there is enough and more of ATP here. So there are a lot of ATPs here. We know that if there is excess ATP, there is inhibition. The TCA cycle will not take place. Why? Because there is enough ATP. It will not take place to that much extent, so that it can use completely that all the acetyl coenzyme A. Apart from that, we know that for the acetyl coenzyme A to enter into TCA cycle, it should combine with oxaloacetate, right? So what happens to this oxaloacetate? As there is excess gluconeogenesis, the oxaloacetate is depleted. So oxaloacetate is depleted. So you can see here oxaloacetate is depleted and there is enough ATP, means the TCA cycle is also not taking place that much, means the major route the acetyl coenzyme A is entering into is for the ketone body synthesis. But remember, it is not taking place inside the in the adipose tissue. This acetyl coenzyme A will go to the liver, and in the liver the acetyl coenzyme A is converted to ketone body. So in the liver we can see is the ketone body synthesis. So this is the basic cause for diabetic ketoacidosis. So which means that there is inhibition of, or there is less amount of glucose uptake by glucose transporter 4. The blood is loaded with glucose, but the cells is giving the signal that no glucose, so there is excess gluconeogenesis. All the substrate for gluconeogenesis will be used up, and insulin generally inhibit the hormone sensitive lipase, but in the absence of insulin there is excess hormone sensitive lipase, so there is excess glycerol and there is excess free fatty acid. This free fatty acid is going to beta oxidation. There is excess acetyl coenzyme A. The acetyl coenzyme A, rather than entering into the TCA cycle, will be going for the ketone body synthesis. This is the metabolic defect in the type 1 diabetes complication, that is diabetic ketoacidosis. So now we'll go back to the question and see what is the answer. Decreased beta oxidation of fatty acid. Is it right? Is it decreased beta oxidation? No, it's not right. So it is false. Increased mobilization of fatty acid from adipose tissue. It is a correct statement. True. Decreased acetyl coenzyme A entering into TCA cycle. Yes, I have shown that. So it is right. True. Increased beta hydroxybutyrate dehydrogenase. What is beta hydroxybutyrate dehydrogenase? This is an enzyme for ketone body synthesis. It is an enzyme for the ketone body synthesis. The beta hydroxybutyrate synthesis, it requires beta hydroxybutyrate dehydrogenase. So that is a true option, which means in the ketosis there is B, C and D. 3 options are correct. So this is answers were like this: A, B, C, D, which is correct. So the answer here is B, C and D. These 3 options are correct. So this is what is the metabolic defect in the diabetic ketoacidosis. Now moving on to the next question. There was a image based question based on the graph. I didn't get actually the correct what was the graph, but from the recall session, recall that is given by the students, what I could understand is it was a graph of the activation energy and the action of enzymes. That is, transition state is reduced, lowering of the transition state by the enzyme. So we will be seeing that options. I don't know whether this is the exact graph which was asked, but I'll just explain it. So you can see that this is a graph of free energy change with the reaction coordinate. So you can see this is the substrate and this is the product. The free energy of the product and this is the free energy of the substrate. You can see that without the enzyme, you can see this is the transition state. This substrate should get this much amount of activation energy. So you can see here this much amount of activation energy if it is getting, the product will be formed. But what happens in the case of the enzymes? The enzymes, if it is acting, you can see the transition energy or transition state is lowered, so that only this much amount of, what you can see here is only this much amount of activation energy is required if there is an enzyme, so that products will be formed easily. So 3 graphs were given, and which of the following graph is showing the enzymatic reaction? So the enzymatic reaction is not shown by A, it is not shown by C. There was a graph like this also. It is not what is an enzymatic reaction. The enzymatic reaction is the graph B. Graph B, where you can see the transition state is lowered, the activation energy required is less. So that is, I don't know exactly what was the question, but roughly a graph like this was asked. So you should be thorough with all the graphs in the enzymes. One more question is, identify the hair abnormality in the polarizing light microscopy. It's an integrated question between biochemistry as well as dermatology. You can see here what you are seeing is hair. In the hair, what you can see is a pattern. This pattern is called as tiger tailing. Tiger tailing. This tiger tailing pattern in the polarized light microscopy is seen in a condition which is called as the TTD. TTD. What is TTD? TTD is trichothiodystrophy. 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 So in this trichothiodystrophy, what you can see is this tiger tailing, and what is this trichothiodystrophy? Trichothiodystrophy is due to a defect in DNA repair mechanism. Defect in DNA repair mechanism. Which repair mechanism? DNA repair mechanism, that is homologous recombination. Homologous recombination defect causes trichothiodystrophy. So way ahead to NEET 2021. The exams are being postponed. Is it a good news? I don't think. Is this the time to relax? No, this is not a time to relax. Then keep going. You are near to the victory. Keep going. Each step may get harder, but don't stop. The view is beautiful at the top. This, about this, I just want to share my experience in my life about postponing of the exam. I have an experience to share when I've written my UG entrance exam. When I did my first UG entrance exam, I was very weak at physics, but chemistry and biology was my strong points. And that exam, all these exams were over, and due to some reasons, the physics exam was postponed because some malpractices were there, etc., etc. So that government has decided to conduct the physics exam again. I got about 1 month time to prepare the physics, which was my weakness. I stay focused during that time, and I tried to make that my weakness, or I tried to strengthen my weakness during that time. Again, the physics exam was conducted, and the second time I could do much better than the first time. That's why I could get a good rank, a decent rank for the UG entrance exam, and I got admission to MBBS. That was one of my experience of postponing the exam. Another experience I had when I was in the final MBBS exam. For the final MBBS exam, model exam, I did well in the sense that I was the topper in my college. I got first place in all the subjects, medicine, surgery, gynae, pediatrics, all the subjects combined. I was the topper. Nobody was there to, nobody was there to cover me. But what happened? For university exam, postponed. So model exam was conducted with only 2 weeks study leave. I was with full energy. I learned for the model exam. I got very good marks. Then the university exam, due to some political reasons or something, that university exam was postponed. Then it was actually, I couldn't stay focused. And all the more, my lover's exam for final MBBS was over. We are a same batch, different college. His exam was over, mine is not over. End of everything. I was totally into phone and everything. I couldn't stay focused. I couldn't study. Then what happened? I was pushed back to the third position from the first position for the university final MBBS exam. So 2 experience I shared. The same person, 2 attitude, which made a huge difference in the outcome. So this is what I want to share. That is, the weakness of the attitude is the weakness. Weakness of attitude becomes the weakness of the character. And Winston Churchill is saying, attitude is a little thing that makes a big difference. So this time is the time you have to stay focused. Your attitude should not be to be relaxed. No, this is not the time for that. You are near to the victory. So this time what you can do? Number one, you can strengthen your weakness. Strong isn't about having a million strength. It's about facing and dealing with your weakness. This is the time where you can strengthen your weakness. That is the first thing that you can do at this time. And very, very important, strengthen your strength, right? So if you are not revising, it means that you may be strong in some subject, or maybe some subject in say certain topics will be a weakness. You have to strengthen your weakness. At the same time, you have to strengthen your strength. And very, very important, see, on the strength of strengths, the weakness weaken. So all your weakness can be covered by your strength. So don't forget to revise all the strong points or the strong subjects, strong topics. How can you revise? Do maximum questions. Don't plunge into the theory portions now, especially for the strong points. Try to do maximum exams in the form of QBank, GTs, or whatever you like. There are a lot of sources now. You can use any of the source and try to do maximum questions in the strong points and make your strength or strengthen your strength so that your weakness can be covered. Because however you tried, the weakness cannot be strengthened, can be strengthened to a limit, but not beyond that. But you can strengthen your strength because that is also equally important. Remember, the storm is a good opportunity for the pine and the cypress to show their strength and stability. This is a stormy time in your life. But this is a time where you can show your strength and stability. So we have to convert the uncertainties to the opportunities. Remember, between every two pines is a doorway to a new world. So don't get weakened during this time. You have to strengthen your weakness and strengthen your strengths. There are a lot of strategies explained by all the faculties. So I don't want to give you a strategy to prepare during this time, but I just told you a few points. What you can do is an inverted cone revision. You can divide your total time into 3 phases. The first phase will take more time. If you have 90 days, then you can divide. First phase will take more time, second phase less time, and you have to even more. You have to make the third phase even more lesser time. So in this first phase or in the second or the third phase, what you can do is some strategy, which is first thing is that you have to find out how much time you have to give for each and every subject. That depends upon the weightage of every subject. Apart from that not only the weightage of the subject, it should be multiplied by the weakness. Some subject, for example, I'm saying about my subject, biochemistry, maybe, when you compare with all other subjects, the number of questions will be 12, 13 or maximum 15 questions. So the number of days that you are giving for biochemistry may be 1 day or 2 days. But that should be multiplied by the factor. What is that factor? Depending upon the weakness, which means that biochemistry, if it is your weak subject, then you have to multiply it by a certain factor so that you give some more time for biochemistry. Likewise, you should divide your days not only depending upon the weightage of marks, but you have to multiply it by a factor, which that factor should be how much weak is that subject for you. So depending on that, you have to divide the days which is available in front of you. That's the first thing that you have to do. So divide the days. So when you divide the days, keep in mind, you have to divide based on the weightage of that subject multiplied by how much weak you are in that subject. Then what you can do is each day if you have 24 hours, out of which if you are learning for 12 hours, say 12 hours, you can set apart 2 to 3 hours or 4 to 5 hours depending upon you, each and every person. Maybe 4 hours you can set apart for your weak subjects. So in a day, if there is 12 hours, 4 hours you set apart for your weak subject, and the rest you can give for the revising your strong point, because that is also very, very important. Because if you are trying to make your strengthen your weakness, don't forget to strengthen your strength as well. Okay, so give that. For that 8 hours you can do maximum questions, but for the weak subjects you can, if you want to see the videos, you can see the revision videos, you can see, or you can if you want to do the Q bank, you can do the Q bank of the weak subjects maximum. So sometimes you can give the night time for your weak subjects and daytime for your strong subject, or reverse also is possible, daytime for your weak subject. So divide that day into 2 parts where you are giving some time for your weak and some time for your strong subject. So likewise you can plan and divide. So depending on that, you have to plan on your first phase, how much time you have to give for the second phase, and how much time you have to give for the third phase. Third phase is very, very critical and the crucial time, because this is the time where you should get time to go through all the topics that you have learned. So that time, that last third phase, how much time you can decide on that. So last phase is the most critical phase in the preparation of a competitive exam. So what I want to give is that inverted cone time, or this inverted cone revision will give you an ice cream. That is your success in the NEET PG exam. So what you have to do is starve your distraction and feed your focus. Stay focused. So just take my life experience, and planning is the key. No one ever plans to fail, but people fail because they simply fail to plan. So you should have a perfect plan, which is a customized plan where you can make your own plan. Nobody else can. I cannot make your plan. It's not possible. I can give some suggestions, and you have to find out what is your plan. And your revision, your hard work, your dedication will bring you success, and all the best.